Hi everyone, today I'm going to guide you through PLTW's slideshow on tissue typing, something that I know only a little bit about, but PLTW does a pretty good job of explaining it, and I hope they can give you a little bit more context. In the last activity, you looked at blood typing as a way for individuals to give blood to one another and hopefully be able to infer that they can give organs to one another. But I'm here to tell you it's a little bit more complicated than that because of the way that our leukocytes are formed. So everyone has several antigens on the surface of his or her leukocytes, meaning that those little white blood cells have these little tiny markers on the outside of them, and they have several different kinds. And of course, they're all different depending on who's who. One particular group of these antigens are called the human leukocyte antigens, the HLAs. Now, the HLA is responsible for stimulating the immune response to recognize tissue as self versus not self, as in, if I have a particular HLA and another piece of tissue comes into my body and does not have that same HLA, the immune response will indicate that as non self and try to destroy it. It's also going to be a specific thing that's controlled by a set of genes located next to each other on chromosome 6. This set of genes is called a major histocompatibility complex, and it's sitting on chromosome 6 right here on the P arm. And of course, we know that the chromosomes have two copies when they're made, so we have really two P arms right here. Now, the test that determines which HLA antigens are present in a person's tissue is called tissue typing or HLA typing. So the test that identifies the similarities of the antigens that are present in both the donor and recipient is what we know as tissue typing. Kind of like blood typing. We know that blood has a certain phenotype that we can match between donor and recipient. Well, tissue typing is going to be a similar test looking specifically for this HLA complex here. The closer that these HLA antigens are on the transplanted organ matching the recipient, as in the more of these HLAs that I can get to have the organ that I'm taking from someone and putting into the patient's body, the more likely that recipient's body will not reject the transplant. It doesn't mean that it won't, but the more HLAs that are on the donor organ that match the patient, the better. For this reason, the tissue typing of the kidney donor and recipient, especially in this activity, is going to be necessary before doing the kidney transplantation. Now, there are two main classes of HLA antigens, class 1 and class 2. I'm not going to make you memorize the abbreviations of all these different things, but they kind of all flow together in the case that every person inherits each of the following antigens from each parent. There's two classes, one for each parent that they get. So HLA-A, B, CW, DR, DQ, and DP are the 6 different types of HLA antigens, and they're all going to be from that spot on that P arm on chromosome 6. Now, the set that we specifically receive from a parent is called a haplotype. Haplotype is just a specific combination of genes specific to one parent going from parent to child. In this case, the haplotype is talking about HLA antigens. There are a variety of alleles for each of these HLA antigens. I could have a different version of the HLA-A allele, the CW allele, and the B, so on and so forth, which means that there's multiple types of haplotypes that are possible for these individuals, for each individual in a family. Because there's a large number of possible variations and combinations of these HLAs, think about it, if there's even just 2 alleles of these 6 different genes, then there's 2 of each, and the combinations would be 2 times 2 times 2 times 2 times 2 times 2. I think that's 32 combinations or something. That makes it very difficult to find a match in a family uh, that is more spread out, like a extended family member. But finding a match in a family is more likely than having a general public individual. And that's why you see so many family members give up kidneys to donate to another individual. When we're performing an HLA typing test for a kidney transplant, the following ones specifically are going to be looked at: the HLA-A, the B, and the DR. The major histocompatibility complex genes are the most polymorphic known, meaning that they have many different forms to them. There's many alleles to them. In fact, there are hundreds of different alleles for each of the HLA antigens. So remember just a second ago when I said there was 2 of each kind, if there was 2 of each kind? Well, really there's more like 100 of each kind. And so that's 100 times 100 times 100 times 100. And so we actually identify each allele by a number. So if I'm talking about the first type of allele for HLA-A, we're going to label it as HLA-A1. HLA-A2 be used for the second one, so on and so forth. So you can see how quickly we could have uh, very many different ones labeled with just the number listed after it. So 6 HLA antigens are looked at for each person being considered for donor and the recipient themselves. Remember that each person has 2 of each of these antigens, one inherited from the mother and one inherited from the father. Hence, if we have this as our mother's haplotype, where they have HLA-A9 and HLA-A3 at these 2 spots for these 2 copies of their chromosomes, and the father has HLA-A29 and HLA-A1, well, the father could pass down HLA-A1 and all this over here, and the mother could pass down HLA-A9 and all this over here, and these could be lost to the inheritance. There'd be no way to get them to the next generation unless they had another kid. But there's a way we can match up the A9 side with the 29 side and the A3 side with a 1 side, so on and so forth. And you get different combinations for each child based on this random flip of a coin of which one I'm going to take from mom and which one I'm going to take from dad. Moving on. So by analyzing which of these 6 HLA antigens both the donor and recipient have, scientists and physicians are able to determine the closeness of the tissue matching, meaning that the 6 antigen match is going to be the best, whereas 0 antigen match is going to be the worst. And this match is going to occur 25% of the time between siblings who have the same mother and father, because there's 4 possibilities. The left chromosome and the right chromosome from mom, the left chromosome and the right chromosome from dad, either or, switching back and forth. It's basically 2 coin flips or a 25% chance. Now, traditionally, HLA typing was done using serological techniques, meaning that the blood from the patient was mixed with serum containing those known antibodies, basically to see if that would coagulate and pool up, just like you did with human body systems back in the day, where you added the different blood types to the pool and they either clumped together or not. And you saw pictures of that on the last activity there. The clumping represents a mismatch of those particular antigens as the antibodies are attacking that particular type of cell. Now, HLA typing can predominantly be done using molecular techniques where we look at the patient's DNA and isolate it, then use PCR to actually amplify specific HLA genes because we know which restriction enzymes can cut at those specific spots. And then the genes are sequenced to determine which alleles are present. So I only have to sequence a very short section, and that specific section is the HLA spot that we looked at before. Now, once the donor and recipient have been tested for tissue compatibility, the next step is a antibody screening, also known as a panel reactive antibody or PRA test. This will incorporate a small amount of the organ recipient's serum, aka the blood that they have with the leukocytes, mixed with cells from 60 different individuals. Each test is going to be done separately. We're not going to put all 60 into one pile and put the serum in there. Instead, we're kind of taking the serum and testing against 60 individuals that we know specifically the combinations of their HLA antigens. The purpose of this is that the scientists can now determine how many different HLA antibodies a patient has in their blood. So if a patient reacts with 30 out of 60 of them, they have 50% of the antibodies possible, and therefore we say that they have a 50% reactive antibody or 50% PRA. The lower that the person's PRA is, the less likely he or she is to reject a transplant because, of course, they don't have as many HLAs that are responding to the 60 individuals that we know. So if there's no reaction, then that means that it's okay for us to put that organ in that body. Now, after the tissue typing and the antibody screening are complete, and a potential donor has been identified, let's say we have an individual who has correctly been identified as a tissue type, and that the antibody screening worked out in both the donor and recipient. The final test is going to be called a cross-match test. The cross-match test is actually exactly what it sounds like. A small amount of the potential donor's white blood cells are going to be mixed with a small amount of the recipient's serum, the blood serum that they have, the plasma, and all the cells that they have. And by exposing this donor's HLA to the recipient's serum, we can actually see if these things are going to mix well, if they're going to play nicely with each other. They can determine if the recipient has antibodies to any of the donor's HLA. And if they do, that's really bad because that will be attacking the donor organ, of course. We want as few as possible. The ideal is 0, but few as possible is the better way. A positive cross-match will have a reaction between the donor and recipient sample, and it indicates that the recipient's body will reject the implanted kidney, whereas the negative cross-match would be no reaction between the two, and that would be if the transplant can be performed and the recipient's body will likely not reject the implanted kidney. Again, we're looking for as likely as possible scenario here. So it's not going to always be perfect. And there are cases where individuals have passed all these 3 tests, but their donor organ still was not accepted by the transplant recipient. But hopefully by the end of these 3 tests, we will know whether or not this donor's organ will be accepted and whether or not it will be attacked by their own immune system here. So that's the basics of tissue typing there. Feel free to watch this video again and make sure that you write some things down from it. 432B will have you looking at Diana and all the different family members that are willing to give up their kidney for Diana and see if there is a way that we can match up the HLAs, excuse me, the HLAs of Diana and the possible donors and see if there's anyone that can actually give their kidney to Diana. Good luck on 432B. Ask questions. If anything doesn't make sense here, be sure to ask me. Good luck. Be smart.